close this.
Hervé is presenting next week, by the way.
Hey, Catherine.
Hello.
How are you doing?
I'm good. How are you?
Good, good,
Thanks for
good.
joining
Hey,
us today.
Hervé.
Hello.
Here we go. The live stream is working. All the speakers are here. Time to start. Thanks for joining Neuroccino, everyone. Great to see you. I hope you had a lovely weekend. We're starting this week with great news, and that is that our YouTube channel has surpassed the 1,000 subscribers. Thank you, everyone, for making that happen. It's quite exciting to see that what we do here every week and once a month is actually appreciated. So thank you very much for that. Right, without further ado, we promised Michel he can go fast after we bumped him last week. So I'm going to hold my promise. Take it away, Michel.
Boom. There we go. Brain pathology recapitulates physiology and network meta-analysis. In Communications Biology, just published last week, also 2 weeks ago. I've been very impressed by this paper. Basically, what they did, they compare resting state functional networks with task-related fMRI, task-related fMRI ICA, so networks that are derived from task-related approach, which has been done previously by Stephen Smith in 2009 in PNS, but they added to that pathologies, neuropsychiatric and neurological disorders. And so what they've been able to show is basically when you do an ICA decomposition of task-related activations, or you do an ICA decomposition of voxel-based morphometry in patients with neurodegenerative disease, you end up with similar networks. Meaning that the different element constituting the network organization of function and damage seems to overlap. So of course here they did it only with 20 components, and I found it pretty clever the way they represented here the same analysis according to decomposition in more actually networks. So here they did 20, here 45 and 70, and they have this gorgeous like 3D representation of how many of the network in percentage were significantly cross-correlated, whether this is VBM, ICA, or functional ICA. Okay, so in some they replicate this finding with 20 components on the left and 20 components on the right, with more components, 45 and 70 components here, and they show that that works even if they go further in the decomposition of the signal. So the first result of the study is really showing that the organization of functions, the way you have different functions of the brain, is the same that the way you have a variance in your gray matter concentration, suggesting that disease progress along functional networks and not across functional networks. Then they try to look at the entropy of disease, which is like, are there some networks that tends to overlap with more disease than others? So by entropy, they mean complexity, which here they're showing that, for example, for the silence, the ventral attention, and the central executive networks, you're more likely to have this network damage, whatever the neurodegenerative or neuropsychiatric disease that you have. While some of the networks, such as the anterior DMN, posterior DMN, or hand sensory motor networks, you have less pathologies that can involve those networks. Okay. Another step of analysis here was to make the link between the different networks they found and the different functions, the different networks that they found and the different pathologies and task-related activation here, which is a useful table if you want to interpret functional networks, or the link between this resting state network and the pathology related to the function. And finally, they do an analysis onto like the level of metabolic use of the brain, and they show that the higher the metabolism in the brain, the more function you will be likely to have in this area, and the more likely you will have different kind of neurodegenerative disease. So it all centered onto high metabolic areas of highly functional and use area in different functions, and the most likely to be damaged across different pathologies of the brain. And that's about it. And I really like this paper because it links up together the functional organization of the brain with its connectivity derived from functional connectivity and a network pathology. And it answers like a question, something that, you know, we were saying, which is you take neurodegenerative disease like Alzheimer, it looks like it's really progressively destroying the episodic memory system. You take another disease like primary progressive aphasia, it looks like it's progressively destroying the language system, as if those diseases were following networks. And this paper is really the one that is demonstrating that. So bravo to the authors, I really enjoyed it.
Lovely, thank you very much. This is indeed an exciting paper. Are there questions in the room? Yes, Leah, I can see you have a question.
Hi, thank you for presenting. And when you were explaining the paper, you remember me about Seeley et al. 2009, I think, when they did kind of the same thing, but using healthy controls for the resting state network analysis, and then compared to the neurodegeneration, cortical atrophy, and so it was the kind of the same parallelism, but here, if I understood well, the fMRI was on the patients too, right?
Yeah. Here you're looking at disease of the brain and how disease follow networks, and different disease actually follow different networks. So you can see here, I'm sorry for the complexity of the table, but there is no way to display better this three-dimensional thing where you have the brain map behavior. This is task-related activation. Here you have the neurological diagnosis, and here you have like the different functional networks. You can see how you have high relationship between those three.
Okay, thank you.
Lovely. Any other questions? Yes.
Yeah, Michelle, did they talk at all about mechanisms for how it could go along networks? I know there's been discussion in Alzheimer's research that maybe prion disease is following networks, you know, through some kind of like facilitated pathway via activation, but I haven't followed up on that in recent years, and I was wondering if they talked about that at all.
So I actually prepared it for last week, so I kind of forgot part of the discussion now, but as far as I remember, so there is this main thing about like disease following networks, but then like the discussion is really centering onto the higher energy cost and metabolism, and how high metabolic area are more fragile or you know, sensitive to disease, and that's why they actually are the most correlated with across disease and have the higher relationship with entropy of disease. But at the center of this is a discussion here.
Cool, thanks.
Lovely. Abby, yeah.
Yes, just can you get back to the figure where it's about the metabolism? I didn't get, yeah, this one I didn't get. So can you go again and see, because there is 3 axes, and it's kind of difficult to get it.
All right, so they look at the level of metabolism used, and here, like the percentage of maximum entropy, which is like how much do you load on different behavior or different disease, different behavior in blue, different disease in orange. So the higher you are, the more pathology will be touched in this area, and the higher you are on the blue, the more domain of function will be touched. And what we see is that the high overlap for pathology or the high overlap for functional domains are well related with a high metabolic cost in the brain.
Okay. Makes sense. Thank you.
Anyone I didn't see? No, I think we're good for now. Thank you very much, Michelle. And thanks for bringing the paper a week later. Very much appreciated. Right, we're going to move on to the next paper, and I'm very pleased to welcome Catherine, who will present for the first time in Neuroccino. That's exciting for us, and quite interesting. We're going to learn everything about the chunk for those of you who followed us on Twitter. Catherine, if you just share your screen.
Sure. And I didn't know how much to prepare, so how long should I talk for?
Just present the paper as in what's interesting, what got you excited, what is
Okay.
it about?
45 minutes.
Okay, good, because I made a lot of slides. I got nervous when Michelle did his. I was like, oh God, was I supposed to talk for 5 minutes? Okay,
It has a
cool.
tendency to make our speakers nervous.
Okay, cool.
But unrelated to how long, if everyone could just share the link so people can follow the publications at their own time as well, and I can copy them over on YouTube as well, so they know what we're looking at. Thank you. If you want to take it away, go for it.
All right. Okay, so we were talking about on Twitter about measures of how chunky a brain is, or how globular, to use these authors' terminology. So Michelle let me know that we would be discussing this paper at Neuroccino, and so I learned about Neuroccino. So thanks so much to Michelle and the rest of you for inviting me to talk. So yeah, this is an interesting paper that to me is interesting because I've been following some of the research on paleoneurology for a few years now, and how it relates to human brain evolution. So in this particular paper, they're interested in understanding sort of the globularity of the human brain. And actually, they don't really define it, but I think basically they mean that human brains are taller and kind of more compressed and rounded than Neanderthal brains and the brains of other hominids. And even if you look at chimpanzees or other primate species, their brains are not as tall and sort of spherical as humans are. So there's been interest in this, and how it came about, why human Homo sapiens evolved this brain shape, and also whether it has behavioral implications and whether it's related to modifications to genes, if there was selective pressure that shaped this, and what that selective pressure was about. So this paper tries to look at all of those things, but really the method that they're using is doing computed tomography to kind of analyze the vertices of the surface of the brain in humans and other hominin ancestors and compare them. So they're really only able to do sort of a morphometric analysis, but they are interested in sort of tying this into other research that looks at genetics, that looks at the emergence of behavioral modernity. So that's the gist of the paper. And they're also especially interested in how this emerged developmentally, and they bring up previous research that seems to indicate that the human brain shape and the novelness of it emerges early. So it seems to occur towards the later weeks of gestation and after birth. So there's kind of a period of brain growth in humans that's characterized by this sort of puffy shape that starts to emerge very early on. And so there's these sort of trajectories where people have tried to map what they think this process looks like in humans, that's here in blue, and in Neanderthals in red here. And so there's some decent evidence already that it emerges early. And another way to look at this, oh, it's covered up my notes, sorry about that, is to look at, to compare the brains in terms of areas that have shrunk and areas that have expanded. So here on the right, you have human brain is on this right hand half, and the Neanderthal brain is on the left half. And so the red is areas that are smaller in that species, and the green is areas that are larger. And what's important to note here in this figure is that it's not just the cranium itself, it's also other components of the skull. So the midface has differences between Neanderthals and Homo sapiens, and the jaw does as well. And one way to kind of visualize this is from a different paper, this Ponce de Leon paper, where they have this nice animation that goes between Neanderthal and human. I kind of wish it was slower, but you get the picture. So it seems sort of like a global change in just the organization of the face and the cranium. And I think what inspired this paper, it appears, is a very early Homo sapiens fossil from 300,000 years ago. And they have what appears to be sort of a Neanderthal style brain organization. You'll notice that the forehead, for example, is very sloped, and the brain appears long. It almost seems to have like an occipital bun, like you might find in a Neanderthal. So they were interested in this because it suggests that essentially this modern human brain shape emerged over the course of evolution of Homo sapiens once the species emerged, and it wasn't something that was what Homo sapiens initially started out with. So they wanted to know, when did this globularity, as they call it, emerge? And so on the right-hand side here, I just have, this is from, I think they're supplemental figures, their comparison of Neanderthals, humans, and Homo erectus, to show you how it appears that other hominins previous to Homo sapiens really were kind of conserving this sort of more sloping brain shape and did not have the human brain shape. And so they were interested in looking at across species, what were the patterns of brain shape, and also across time. And I think basically their hypothesis is that both across species and across time, you see a change over to this more globular brain shape. And they use principal components analysis to look at this, look at these. And let me see, yeah, sorry. And I should mention, they looked at Homo sapiens fossils, and from starting as early as the Moroccan specimen that they mentioned, all the way to more recent ones that look very, very modern. They have 8 Homo erectus, 8 Homo neanderthalensis. They had 2 Homo heidelbergensis, although it looks like species designation is a little bit disputed in the literature. I don't have any comments on that. I don't know if anybody here is, if this is their field. So if they do, please chime in. And then they have a lot of human samples. So they have 89 modern or present day human samples, and they selected from a wide variety of locations. So they have a really diverse human sample, which I think is really good. So here's their analysis of variation of total endocranial shape. So one important thing to note here is they registered them all to the same size. They corrected them to the same size because they didn't want size to be impacting this principal components analysis. They just wanted the shape. And they identify kind of, so in the yellow is Homo erectus, the red is Neanderthals, and the blue are different humans at different geologic ages. So this triangle here, 1 is the earliest geologic age, and then 2 is the middle one, and 3 is the more recent 10,000 years ago Homo sapiens. And then as you see, it overlaps with their present day human samples. So they're showing there's this movement along principal component axis 1 in humans that we're not seeing in these other, that's a novel movement in sort of shape space that we don't see in these other species. And they're arguing that basically the modern day human brain shape variation reached sometime between 100,000 and 35,000 years ago. Now that's a really big, it's a broad time period, but that, that's what their data support at this point. And they're, to be more specific, it's this geologic age is explaining 21% of, or 22% of shape variance. Another important thing to note is that they did not find any geographic component of the shape variation. So it seemed to be, mainly driven by time. And this is just another way to visualize this pattern of change that they're seeing, where essentially the brain is getting taller and narrower, and the cerebellum is kind of getting pushed more forward, I would say, and underneath the brain. So this is just in their supplemental. They had these really nice sort of visualizations of what's happening over geologic time in Homo sapiens. So I thought those were worth looking at. And I think it really helps too, because when you say globular, what do you mean? And they talk later in the paper, they're really talking about the sort of increase in the height that seems to be especially focused around the parietal areas. And you kind of also have these temporal lobes that kind of push forward. To help you get a visualization of what exactly is happening. Oh. All right. Here they did the same analysis again, and from what I can understand, they just, in this time, didn't remove the shape, but didn't remove the size variable. They also included size and shape. I don't know, honestly, to me, this made it a little more confusing. I think what they were trying to demonstrate here is that size and shape sort of had different patterns of change. And I think they wanted to illustrate that size was not playing any kind of role in this change in shape, and just show it in a different way. And also show that Neanderthals kept increasing the size of their brain, but they completely maintained the original sort of archaic shape that we found in other hominins. So I think that's pretty interesting. And again, here, geologic age is explaining roughly the same percentage of shape variance that we saw in the previous analysis. So I guess you could say that's helpful. And yet in archaic Homo, we don't see a similar change that corresponds to geologic age. Actually, I'm going to skip this because I think this is actually a little bit confusing. Okay, so to be more specific about what these shape changes look like, it mainly seems to focus on the sort of anterior and middle cranial fossa. So, and they want to also be clear that this globularization is occurring after we've achieved essentially modern day or present day Homo sapiens brain size. So they're kind of arguing there's two-step change. There's a change in the increase in brain size that we see in Homo sapiens, but then there's also the second step change, which is the change in shape. And so they mentioned this anterior and middle cranial fossa seem to be the main place where there's sort of a change in the geometry. And they did briefly discuss the fact that this may reflect a response to changes in the face. And I thought that was interesting because that's something that other authors have talked about, especially I think Emiliana Brunner has talked about it. I know Bastyr has talked about this quite a bit, that you can see this very nice correlation between as the midface sort of retracts and that you see a corresponding more globular Homo sapiens brain. And so for some paleoneurologists, I think they might argue this is really driven by face changes and jaw changes, and that it's not driven by cognitive factors. And we obviously can't test that, but it's interesting. These authors, I think, are not as compelled by that argument, and they want to talk a little bit about what areas appear to be larger and taller, and what that might implicate. And so their argument is, well, we really are seeing these parietal bulging changes and these cerebellar changes. And so they talk a little bit about, what the parietal lobes are for, which I think most of us know, which has to do with the dorsal visual pathway and visuospatial processing. It's also involved in a lot of pathways that connect to the prefrontal cortex that are involved with language and tool use. So you could argue, especially tool use, that, perhaps in this period of human evolution, there may have been selection for abilities that required parietal specific processes. And they talk a little bit about the cerebellum, and I'm not sure how to connect that to this. But I think one of the more compelling things and I think they're right about, is there are shape changes in these areas that are unique to humans that are worth looking at. And I'm thinking specifically of the precuneus, which is something Emiliana Brunner has worked at, showing that there's a lot of variation. A, the precuneus in humans is quite large, and B, there's a lot of variation in humans, which may suggest that there was a recent selection pressure that's not stabilized yet for a larger sort of medial side of these parietal bones, like the structures in the medial component of that. So I think that's pretty cool. They also talk a little bit about what this might mean for looking back at the fossil record in terms of tool use and other behaviors, and can we find some kind of connection between when the step change was happening, when this globularization was happening, and the emergence of what people call behavioral modernity. And this isn't my area of expertise, so I'm happy to if people here have things to say about it. I looked at some of the references they referred to around this, because their argument is essentially this is a good possibility for explaining the emergence of behavioral modernity in Homo sapiens, because they're sort of saying in this last 100,000 years, when we see this globular brain emerge, is when we see major components of human behavioral modernity. And some people, so what is behavioral modernity? I'm not quite sure, but it has to do with the emergence of new tool technologies, more refined tool technologies and lithics. And one of the authors mentions, several other things like more travel, possibly trading between human populations. Let's see, what else? Processing like food and other objects more, using pigment, etc. I think this is a difficult argument to make. And one of the citations they used actually argued that many of these behaviors are seen in hominin ancestors and are not unique to Homo sapiens, and that it's not really fair to say that behavioral modernity is a suite of things that just happen like that, and that's how modern humans emerged. So I think it's kind of I think it's difficult to claim this, but it is interesting. And I could see, for example, I would be interested to see someone analyze the changes in globularity and maybe look specifically at tools, because that is at least something that we have like a consistent fossil record of. And we could talk about the emergence of different tool assemblages. It gets a little tricky because we know Neanderthals did some of these tool use, tool manufacturing technologies as well. And there's debate sometimes about what tools belong to which group, but maybe there's something there that could work. So essentially their main conclusion is that this is a directional gradual change to increased globularity in the evolution and emergence of modern day Homo sapiens. And they encourage people to start looking more at how this connects to behavioral modernity, and however you might define that, and how it may connect to changes in brain development. And they make a pretty good case for looking at the genetics of brain development and how selection may have been shaped in human brains, and we might find some genetic markers of this that have led to these changes into a globular shape, which I think is really interesting. And I would also be interested personally to see if people could also look for genetic markers of maybe facial development to try to get a handle on, is this driven by a cognitive mechanism, or is this driven by some other kind of mechanical or other selection pressure that has to do with the face, whether it has to do with chewing or does it have to do with breathing? I don't know, but it was a lot of food for thought. So I thought it was a really interesting paper. So yeah, that's what I have.
Lovely. Thank you very much. Super exciting indeed. And thanks for preparing a proper presentation for us. Personally, I thought the videos were absolutely stunning too.
Yeah.
Really see the evolution from one to the other.
Yeah.
Right. I can already see questions in the room. So, Michelle.
Thank you. I love it so much, really. And thank you for taking the time, like making all those slides. It really helps to understand. That was wonderful. I have so many questions. I'll try to focus on I want to discuss so many things with you, but I'd like to focus just on the main one, which is, so they use a principal component analysis to simplify a little bit the change of shape and have a point of comparisons across the different human lineage species. I don't know how we're gonna call it. And so when they did that first, is there like some form of like triangles everybody's gonna love that are appearing, so that there is a form of trade-off between traits, so you cannot have a brain that have too much that shape or not? Did they try to look at how, so many questions, sorry, how each axis of components relate to specific things in the shape?
No,
Did they try to do 3 components and see in 3D how does it distribute? Sorry.
No, that's a really good question, and I didn't see it in the paper. Now I'm wondering if it's buried in the supplemental, because I don't know how they selected just 2. Yeah, so I think that's a really good question.
Then they could have done like a regression on each axis and see a little bit whether it's like, you know, the lateral bulging or like more like that is related to one component or another, just to help us have a better interpretation of like what those axes represent.
Yeah.
You know, just a linear regression that would be so great.
Be helpful. Yeah.
Yeah.
It's a good question.
It is so cool. As the data available, can we play with it?
Oh, that's a good question. I wonder if they...
I don't think they make it available.
They should. Especially with the really nice modern day human data set.
All right. That's about it. I'm gonna, do anybody has other questions, or can I carry on with?
But we actually did have a bet on you picking up on the triangles.
I have 2. Triangles are the best. Oh, Leah has a question.
Yeah, take it away.
Thank you. Yes, we see the triangles. Now I would thank you. I really love this animation, and we talk a lot about the parietal lobe. I was thinking we don't have any correlations with the frontal one. Like there is an expansion there, or it's just the parietal?
There is a inferior to superior expansion you can see in this image in the frontal cortex as well. I think the biggest change is in the parietal lobe, which is why they focus on that so much. But for sure. But the other complicated thing is that this sort of geometric expansion is not related to a volumetric expansion. So it's just simply a shape change and not a volume change.
Okay. Thank you.
Any other questions? Oh, Abby, yeah.
It's kind of question and comment. I think the figure 2 with the principal component 1 is one of the most interesting. Not this, this one too, but the first one is really. There is kind of a gap between. So the problem with this kind of data, they don't have a lot for the first group.
Yeah,
they seems to have a gap between the. So the 2 and the 3 seems to be very close, but the 1 seems to be kind of dispersed and far away from the other 2. So difficult to. So it would be better to have more subjects, but
yeah, absolutely. It almost makes it look like it was. It makes it look like, from an evolutionary perspective, that it was perhaps a rather strong selection pressure that made sort of this jump from these 2 different states.
And yeah, I was thinking, because we discussed about the paper, so I looked at, I discussed a paper about the genetics. They in this paper, I don't know the, yeah, it's Underco, I think, Underco 2021. They discussed about the age of the genetic variation along the human lineage, and they kind of try to find gaps between, or not gaps, but period of time where they got a lot of changes in terms of genetics. So I don't know if this kind of changes or big period of changes can be described or find in this kind of analysis. So it will be interesting to see if that. So this is an evolution, but is it linear or kind of bumpy?
Right.
We don't know.
Yeah, and I think let's see if I can I had it up here. I thought also this difference in developmental shape in Neanderthals and modern humans was pretty striking, and I think that's kind of what they're pointing to, is saying we're sort of expecting to see some kind of link for selective pressure, a genetic component to that, which is pretty interesting. I also, like, sometimes I just wonder, how much, brains are pretty flexible, I think, in terms of dealing with deformed crania. So, you wonder how much of this is really about other things, about just bone structure that are driving this. But
yeah, because when you look at the regions, the most different region is the cerebellum, which is a very great region. And so, but always when we talk about evolution, it's about the frontal part and stuff. But when you look at this picture, the biggest changes are in the cerebellum. So kind of interesting.
Yeah, it's always like, I don't know what to make of that. I mean, you obviously need one, but I don't.
Yes.
Beyond that.
So we actually had a question about the cerebellum over on YouTube, and that is whether the cerebellum is associated with working memory.
That's a great question.
Yes, it is. And you can have a lesion in the cerebellum and have working memory impairment.
Thanks, Michelle.
Don't have the reference handy, but I can put it on YouTube later if you want.
Yeah, and it's one of those regions that we initially said, well, it's motor, it's important for motor processing and timing especially, but more and more it's being implicated in language and all kinds of complex cognitive abilities. So
I had a question regarding, like, that's really an open question, but the shape of the skull might be also changing due to customs, where these people holding stuff on their head, making the skull like a lot flatter. We see it, my daughter, she used to sleep, she had a stiff neck, so she slept all the time on the same side, and she started to have a flat bone on the side of her head that we had to adjust, with doctors. And this happened, and there's also this culture where they used to wrap their head and deform their skull to expand their mind, and this also has an impact on the shape of the skull, and must have we're not phrenologists, but it must have a lot of impact on the shape of the brain at some point.
Yep.
I don't know.
And it made me think too about I know that there's been work in modern hunter-gatherer populations about the way they use knives, and they tend to put food in their mouth and then cut it from their mouth, and it has implications on the occlusion of their teeth compared to industrialized people. And I wonder, as they were starting to get these more complex tool assemblages, were they changing how they were processing food, and did that have impacts on this jaw and this mid-face?
Let's go visit those tribes that wear stuff on their head and scan them.
Yeah.
And check whether.
Yeah. So Michelle, I don't know if you knew. I was always interested in that. So I put a link where you can see some tribes in Anka. They used to put stones on their skull from very early in age, And so you can see in the pictures the shape they got with this kind of deformation. It's kind of impressive. I don't know if you can see, but it's kind of, yeah, if you can share, yeah. This is really alien shape, and so I was wondering, so you can do that, but we can do that, but I don't know for this kind of person if the cognitive changes they imply doing that. So when you see the occipital part is completely, the skull is completely elongated. So I don't know how the functional connectivity is working on this kind of case, but it's really interesting. It will be good to redo that and to see if there is implication on that.
I mean, I've seen Indiana Jones, and there are strong evidence that these
yes,
these people are actually aliens. It's okay.
Yeah, I don't understand even how your fasciculi would be organized with that really extreme one. I think it's just mind blowing.
It's yeah, it's good comment. We should put them in an MRI scan. No kidding.
We might have to wait until we have a portable MRI.
Yeah.
And a tailored coil.
It'll be cheaper to pay a flight ticket.
Right. Are there any other questions, comments? No? Okay. In that case, thank you very much for presenting. Very exciting debut on the channel.
Thank you for having me.
Anytime. You're welcome to come back anytime.
Thank you.
Right, with that, let me just point you to the fact that we have a wonderful talk coming up this week from Sophie Park on Thursday at 4 p.m. Paris time, that is, on shaping the brain structure and function. So we hope we see you on Thursday for that one. Other than that, have an exciting week in science, and we see you again next Monday for Neuroccino. And I think Abby is presenting next week. I may have talked him into it. Lovely. Have a wonderful week. See you